Hello everybody and welcome to Prokaryotes and Viruses. This chapter tends to be a little bit long as far as the PowerPoint slide goes, but there's a lot of extra information in here for you, so I'm going to try to pare it down as we go through. So microorganisms in general are single-celled organisms that are too small to be seen with the naked eye. They include bacteria, viruses, protists, a whole bunch of different things. The smallest living organisms are going to be bacteria. They're teeny tiny. They're like hundreds or thousands of times smaller than protists, like amoebas. And thousands of times smaller than any cell within your body. So they're itty bitty, really small. Viruses are even smaller than those, thousands and thousands of times smaller. But they're not considered to be alive. Right? One of the main reasons is they don't have a cell membrane. They are not able to reproduce on their own. They have to actually infect something else in order to steal its machinery and actually reproduce. So they're not considered to be living organisms, which is an interesting kind of deal. Prokaryotes are only two main groups of prokaryotes, and they're set up into two large domains. So the domains are even above kingdoms. They're the largest of the categories that we have. We have domain archaea, and we have domain bacteria, all right, which are archaeobacteria and eubacteria. So each domain associates actually directly with the kingdom underneath. So domain archaea goes with archaeobacteria kingdom, and domain bacteria goes with the eubacteria kingdom. And they're evolutionary, very primitive, right? These are prokaryotes. Prokaryotes mean they're the first cells, and they're your prototypes. They're kind of the basic cells. They don't have a nucleus. They don't have any membrane-bound organelles. Their chromosomes are in a circle, not linear like ours. So very primitive, very basic organisms. So no membrane-bound nucleus, single chromosome in a circle. Cell walls found in most species, almost all of them. They reproduce by prokaryotic fission, which is kind of like mitosis, but simplified down because they don't have all the other organelles and stuff to worry about. And they have a huge metabolic diversity. Right? You're talking about all kinds of different ways of getting nutrients from the world. They've been around for literally billions of years, so they've had lots of time to evolve, adapt, and be able to utilize just about anything and everything they possibly can on this planet in order to get out energy. Here's a nice little picture of a typical bacteria. You have the outside capsule, which is kind of like their rubber coating on the outside to help protect and provide support. Cell wall underneath that, cell membrane underneath of that, and then you have the circular chromosome here that's kind of all spaghettied up. And they do have some ribosomes, but those don't have membranes, right? But those ribosomes help to make the proteins. And they also have a long flagella. It's a very simplified flagella. It's not the same as the ones we have in like sperm or other flagellates, protists, and things like that. But it's at least works similarly, but it's very much simplified. You have three main shapes to bacteria. You have coccus, you have bacillus, and you have spirillum. Coccus is spherical. Bacillus is rod-shaped, long and skinny. Spirillum is spiral-shaped. You need to make sure you know each of those. All right, there's an example. There's your coccus, there's your bacillus, and there's your spirillum, as far as actually looking at what they look like. The cell wall is semi-rigid but permeable, so it still allows things to go in and out, but it provides support. It surrounds the cell membrane. It helps to maintain the cell's general shape and resist rupturing. And it's made out of a very important structure called peptidoglycan, a very important protein. So please make sure you know peptidoglycan. It's found within the cell wall. It'll come up a lot here in a little bit. All right. Gram staining is exactly where it comes into. So gram staining in the cell wall is a useful diagnostic tool. So it's literally a stain that we can apply to the bacteria. And if the stain sticks to the bacteria, it tells us some things. If it doesn't stick to the bacteria, it tells us something different. Right, so it helps us to identify certain types of bacteria and their chemical and physical properties of their cell wall. So you can take an unknown species, you can expose it to the purple dye of the gram stain, and then to iodine. Right, and then you wash it all off, and lastly you do a counter stain in case the stain didn't work. You can stain it with safranin, so it turns in kind of a pink color. So it's going to be purple color, that means it's gram positive. If it's going to be a pink color, that means it's gram negative. That means it didn't take the original purple dye, and the second stain is the one that's stuck is the one that's on there. All right, so gram positive stays the purple, so you can see gram positive coccus right over here. And then if it doesn't, it turns pink, and this is that gram negative over here with the bacillus bacteria. They also have a glycocalyx on the outside, which is like the sticky capsule, right? It closes the cell wall, and it helps to attach them to all kinds of things that they grow on your rocks, on teeth, on intestinal, vaginal walls, all over the place. So pretty much wherever they go, they use this to basically lock them in onto where they're growing so they don't wash away. And when you have the bacteria growing on your teeth, it's not just good to sit there and take your finger across your teeth. That's not going to remove them. You need an abrasive, you need a toothbrush, all that good stuff to get the bacteria away because of this glycocalyx. They have that special bacterial flagella I told you about. Modal structure helps them to propel themselves through any kind of liquid that they're in. And they're structurally very simple flagella, though, not as complicated as the ones you see in eukaryotes. They have a pilus, and specifically the one you need to know is the sex pilus. Right? The sex pilus helps them to connect with another bacteria. You know, bacteria don't actually have sex, but they are able to actually pass a little bit of DNA in between themselves through something called a plasmid. We'll talk a lot more about this here in a few slides. But here's a picture of that sex pilus going through and connecting to two types of bacteria. Prokaryotes exhibit the most metabolic diversity compared to other groups. Right? They've been around for billions of years, and so therefore you have lots of different possibilities. You have photoautotrophs, which means they photosynthesize to make their energy. Chemoautotrophs, which means they use chemicals to make their energy, like the bacteria at hydrothermal vents at the bottom of the ocean. You have photoheterotrophs that still use light, but they actually have to get their energy from another organic source. They have to get their carbon from an organic source. And then you have chemoheterotrophs, which are like us. They have to eat and get all their carbon from an organic source. Photoautotrophs, photosynthesizers, again, carbon from carbon dioxide, energy from the sun. Chemoautotrophs obtain their carbon from carbon dioxide, but they can get all their nutrients and chemical reactions from either manganese, sulfur, nitrogen, all kinds of different things. A lot of these are archaea bacteria. Photoheterotrophs are not so they're photosynthetic, but they can't self-feed. They have to get the carbon from some other organic compounds. They still have to eat something. They just use light to help them get some extra, help them with their process of breaking it down and producing energy. And then chemoheterotrophs, the most common, not self-feeders. They must ingest organic building blocks. A lot of them are parasitic, so they get their nutrients from the host that they attach onto. And then you have some that are saprobic, which means they break down dead and decaying material. They eat dead stuff. These are the ones that make your food go bad, and they'll break down all the trees and leaves and things in the forest, and they'll decompose bodies and all that kind of stuff. All right, so let's look at some of the growth and reproduction patterns. All right, so prokaryotic growth is how they basically grow across a medium. All right, so increase their component parts between divisions, and a population is increasing their numbers. And you can see a growth pattern here on a petri dish down at the bottom right. With bacterial genes, bacteria have a single chromosome, a circular chromosome, right? That's one large circular chromosome. Many bacteria also have little plasmids, which are little tiny little fragments of DNA in a circular structure also. And these can self-replicate, they can duplicate themselves, and they are what are passed in between from bacteria to bacteria in conjugation. Right, and here's prokaryotic fission. Just know that they can do it. They divide, they split, and this is how it's done if you want a little bit more information. Now, conjugation, you got to know. All right, conjugation is a huge thing between bacteria. It makes them extremely adaptable and very efficient, and helps them do a lot of things that cause us a lot of problems. So plasmids can be inherited. So dividing, each cell will pick up that plasmid, but it also can be passed from one bacteria to another. And so it's a mode of gene transfer. One cell has a certain type of plasmid, and it can donate that particular type of plasmid to another cell through that sex pilus. Right? So it can actually connect from one bacteria to another, and then it can donate its plasmid over to that other type of bacteria. Now, crazy thing is, this what's on the plasmid can be a bunch of random genes. It can be a whole bunch of different things that can be on that plasmid. The other thing is it doesn't have to be between the same species of bacteria. They can actually pass DNA and genes from one species to another. So that means a completely harmless species of bacteria can take a gene that allows them to be resistant to a bunch of different antibiotics and pass it over to a very virulent or very nasty bacteria that doesn't have that yet. And all of a sudden, that new nasty bacteria now has the ability to be immune to all those types of antibiotics also. So this is a big deal. This is how we have a lot of problems now with antibiotic resistance being passed along, because it doesn't just have to be nasty bacteria that get those. It can be other bacteria that are common all over the place, and they can build up those same antibiotic resistances and then pass them along to a much more aggressive type of bacteria that does cause problems. So this is one of the reasons why we're having such a big issue with antibiotic resistance, is because not only are they able to go from one to another within the species, but they can actually pass along between species. And this is kind of how it goes. You got your little circular plasmid. They make a copy on the outside, and then they literally send it over by that sex pilus over to the other bacteria. Now that other bacteria has a plasmid with that same information, and on that can be, like I said, a bunch of different things. It can be new ways of getting energy. It can be antibiotic resistance. It can be a bunch of different things. And then if they find it really important enough, they can actually incorporate that plasmid into their circular chromosome, and where it stays permanently. Now, classifying bacteria, like we said, we have eubacteria and archaeobacteria. And archaeobacteria are actually more closely related to us as eukaryotes than they are back to eubacteria. Right? They've been separated out over billions of years, and we've actually, through DNA, have figured out that we're actually more closely related to archaeobacteria than we are to eubacteria. The methods of classifying an unidentified microbe. All right, these are the main ones you need to know. I will ask you this repeatedly throughout the entire year, so please make sure you know these. First one is the shape. All right, I want you to know that you look for the shape. So if a doctor is basically does a swab out of the back of your throat, puts it under the microscope, the first thing you're going to look at is the shape. Is it coccus, bacillus, or spirilla? All right, have you ever heard the term streptococcus? All right, that's because that particular bacteria grows in a form that's strepto, which means in a chain, and then it's coccus, it's round in shape. So if he sees a bunch of bacteria that are long chains of circular or spherical bacteria, then he knows you have strep throat when he does a swab, and that's how they diagnose it, or at least one of the ways that they help to identify it, first of all. All right, so they have streptococcus, or the shape is one of the main factors. The other one is the gram staining. All right, are they gram positive or gram negative? It tells a lot about what kind of antibiotics might work against that bacteria. And the other one I want you to know is how they grow. So we have motility and metabolism, but I want you to know how they grow. All right, how they grow in a petri dish and how they grow on their own. All right, so shape, gram staining, and how they grow are the three answers that I'm looking for on the quizzes and the tests that you're going to have. This will be a short answer question. You will have to do it throughout the entire year because I want you to make sure that you understand this. All right, so they look at the shape, they look at the gram stain, and they look at how it grows. Okay, so make sure you have that. So comparative biochemistry tells us more accurate tales. So this is looking at the DNA. Right, so doing our gene sequencing, and it tells us how certain bacteria have diverged, how groups have come about. It gives us a lot more information on what's related to what. And so again, we have our little family tree here where bacteria broke off, and then over here we have archaea, and archaea is closely related to eukaryotes, more closely related than bacteria is. All right, domain bacteria. Let's look more closely. This is the most ancient lineage of prokaryotic cells. But it includes the most familiar bacteria. It's typically the bacteria that we think of when we think of bacteria. Most of them have a cell wall, and it always includes peptidoglycan. It just depends on how much peptidoglycan is in there. Classification based largely on their metabolism, how they grow, what they eat, how they feed, all that kind of good stuff. And many of these species are parasitic or pathogenic, so they can cause disease or live off of other organisms. You can have photoautotrophs that are aerobic, so meaning they need oxygen, like cyanobacteria. You can have anaerobic ones that don't like to have oxygen, that'll die in oxygen, like certain types of green bacteria. You can have photoheterotrophs we talked about. You can have chemoheterotrophs, like the ones for the nitrogen cycle, or the ones at the bottom of the hydrothermal vents at the bottom of the ocean. And then you have your largest group and your most common group, the chemoheterotrophs, the ones that have to get their energy off of some other living organism. And here's a nice little diagram if you want to look at all the different groups, but not necessary. All right, there's some examples of gram positives. Includes Heliobacter, which causes ulcers, Streptococcus. You have chlamydia, bunches of Escherichia coli. All of those, Salmonella, are all within that group of gram positives. Well, all of these are within gram positives. Gram negatives are down here. Those are Salmonella and White pestis and Rickettsia and a bunch of those guys. All right, so you can kind of see just some examples and kind of where they fall in. You do not need to know those. All right, cyanobacteria are probably one of the most important bacteria that we have, especially the photoautotrophs. They are the first to use a non-cyclic pathway of photosynthesis, and it basically produces tons and tons of oxygen. This is why oxygen came to our planet. They believe that we got so much oxygen. It's because these cyanobacteria start going crazy, photosynthesizing like nuts, and produce tons and tons of oxygen, which filled our atmosphere. At one point in time, we almost had 40 or 50% oxygen in our atmosphere. Now we only have 20%. And this allowed for all kinds of organisms to move on the land and all kinds of other advancements to happen. So it's a very important organism. It's still a very important organism as far as producing oxygen and also utilizing carbon dioxide out of our atmosphere for climate change. We also have these involved in several different symbiotic relationships with fungus to create things called lichens. Lichens are that symbiotic relationship that can pretty much live anywhere. The fungus provides it a nice, safe place to live so it doesn't dry out, and then the cyanobacteria end up producing enough nutrients for both the cyanobacteria and the fungus, and so they create this symbiotic relationship that can pretty much live on bare rock. So a really neat advantageous relationship there. Proteobacteria are gram-negative, so you got some examples here, E. coli, Salmonella, cholera, rickettsia, also known as Rocky Mountain spotted fever. All of these are in this gram-negative category. Chlamydia, and spirochetes, several of them like syphilis and Lyme disease. Right, gram-positives are, most of them are chemoheterotrophs. You got lactobacillus, you got streptococcus, staphylococcus, a lot of spore producers in this group. Clostridium, like anthrax and tetanus and botulism, are all in this category also. Some of the most nasty are the ones that produce some of the most nasty toxins that we know of. And a lot of these can produce something called an endospore whenever they get in a very hostile environment. Right? And this endospore is basically like creating a seed or a capsule around them that can survive extreme heat, extreme irradiation, drying out, acids and disinfectants, doesn't matter how much Lysol you put down, boiling water, all kinds of things. They can live and survive through all these really nasty things, and then when it finally lands in a habitat that's suitable, it will open back up and start reproducing again. So this allows them to survive in some places you never thought they would, and then also can create the problems. This is where some of those just random cases of anthrax come out of the woodwork, is because people are digging or around certain areas where an anthrax bacteria has actually been hiding out for a while, and in a endospore it goes inside the human body by breathing it, because they've been digging in the dirt or doing something along those lines, and then they get infected when it breaks out of the endospore. Same thing with tetanus. Tetanus forms an endospore on the end of rusty areas, on nails and on metal, things like that, and this is how you can get tetanus from a rusty nail. And there's a nice little picture of it. You got the actual bacteria inside and the endospore capsule on the outside. That'll protect it from all kinds of stuff and survive for a very, very, very long time in very nasty environments. All right, let's go to domain archaea. Main thing about domain archaea is that they are the extremophiles. Basically, they live in all the crazy places you never think anything could possibly survive. They're a lot like normal bacteria, or like eubacteria. They're prokaryotic. They're the same general size, a lot of the same types of shapes. But their cell wall does not have peptidoglycan. Instead, it has something called pseudopeptidoglycan. It has polysaccharides in it, glycoproteins in it, several other different things also. And they're more closely related to eukaryotes again than they are to prokaryotes. Right? We got nice little categories there. You can kind of see some examples. Right? The three main groups are methanogens, extreme halophiles, and extreme thermophiles. You must know these. Methanogens make methane. Right? This is what makes you fart. Extreme halophiles love salt, so they're in the Dead Sea and the Great Salt Lake in Utah, all those kinds of areas. And then the extreme thermophiles, which love heat. All right, and so they're in the hot areas like the hydrothermal vents and the geysers of Yellowstone National Park and places like that. All right, so you can have, so these are our three main kind of categories for archaea. All right, the methanogens are strict anaerobes. They die with oxygen. They're found in swamps, digestive tract of animals, hence the farting, and at the hydrothermal vents also. Right, so are all within this category. They have a major impact on global cycling of carbon and the production of methane. Extreme halophiles live in the salt ponds like the Great Salt Lake and the Dead Sea. Like I said here, you've got a really cool picture here in the middle of what they do out in Asia. In a lot of places, they actually have these large kind of areas that they flood with salt water, and they put a barrier around it, so they flood the entire area with salt water, but only like 6 inches or a foot of salt water. And then they let it slowly evaporate. And as it gets saltier and saltier and saltier, it creates a better habitat for these extreme halophiles, and they'll grow and photosynthesize in there and thrive in there until it completely dries out. And then when it completely dries out, they go through and they bulldoze it, and this is how you get your sea salt. And you can see the pretty colors that are associated with these extreme halophiles that are photosynthesizing. And our extreme thermophiles. And they like extremely high temperatures. They like the sulfur-rich hot springs in Yellowstone National Park, the deep sea vents, which can go over 110 degrees Celsius, so well above the boiling. And these guys can survive there. So all the places you think nothing could possibly live, that's where these guys like to live. Lastly, let's talk about some things that are not alive, but still cause infections. They're important to know about and important to talk about. But remember, viruses and everything we're going to talk about from now on are not technically alive. Right, so viruses are non-cellular infectious agents. They're protein coat, right, wrapped around a nucleic acid core. So they have some kind of nucleic acid in the center, either DNA or RNA, and then they have a protein shell on the outside, a protein coat on the outside. And that is the structure of a virus. That's all there is to it. There is no cell membrane, there are no organelles, because there is no metabolism. They are not alive. They can only reproduce with a host. And here's a couple of pictures of one. So you got a helical virus over here, a polyhedral virus, and then over on the right-hand side is probably the most famous type of virus, called a bacteriophage. All right, the viruses on the left are ones that tend to try to trick cells, so they'll attach onto the outside of a cell and trick that cell to bring the entire virus inside. Whereas a bacteriophage grabs on with these arms like a giant alien, injects with these huge spikes into the bacteria, and then squeezes and injects its DNA or RNA into that bacteria and takes over control of it. Luckily, these only attack bacteria. All right, we do not have these type of viruses attacking our cells. Instead, we have the ones that trick our cells. And again, these are very, very small, so all it has to do is trick your cell, and it can slide right in and then start taking control. All right, here's a bunch of different types of viruses. You can see some of the diseases that they come up with: smallpox, cowpox, herpes, chickenpox, hepatitis, all of those types of things. And there's some RNA. So the ones before were our DNA and our DNA viruses. So these are the ones that use DNA, and then these are ones that use RNA. All right, polio, the cold, the flu. You have rabies, hemorrhagic fever, Ebola, measles, mumps, encephalitis, AIDS. All of these are viruses that use RNA. So they don't even have any DNA. All they have is RNA, and they inject that RNA inside the cell, and that RNA actually takes control of that cell. Here's some examples. You can see viruses actually allow and elaborate the color on this particular tulip. You can see some electron microscopy of some viruses attacking certain types of cells. So here's a cell with bacteria all around the outside. Here's a very close-up showing you they're injecting their DNA or RNA inside of that cell, the bacteriophage. And so viral multiplication, you have a bunch of different parts. First part is attaching. So they have to attach to a host. Some of these viruses are very specific on what cells they attach to. They have to find a very specific attachment point. And if they don't have that attachment point, they won't attack the cell. You can have, and then they have to trick that cell to get themselves inside or inject their DNA. So you have some kind of penetration to enter the host. And then they start their replication and synthesis process. So how they actually start affecting the cell, taking over control of the cell, and how they end up doing their thing. All right, they create the assembly or the production of more viruses, and finally release all of those viruses into the body again in order to spread along. We have two different pathways that viruses use. They utilize the lytic pathway and the lysogenic pathway. The lytic pathway is the rapid viral replication pathway. It ends with the lysis of the cell. Lysis means to rupture or to explode. All right, and so there's the lysogenic pathway is all characterized by a latent period. It does not kill the cell outright to start with. It hides inside the DNA, and it waits. So these are viruses that tend to have a latency period like HIV that can hang around in the body for a long period of time before they come out and then kill the host. For the lytic pathway, you have the bacteria that gets injected with its DNA here, but it can be any cell that gets attacked by the virus. The virus puts its DNA or RNA inside the cell. That DNA and RNA literally takes control of the cell. It uses the ribosomes, it uses the endoplasmic reticulum, the Golgi, all the different structures of that cell, takes over control of them and makes them produce more virus parts. They produce more virus components, more virus DNA or RNA, depending on what type of virus, and completely construct new viruses. Those new viruses build up so much inside of that cell that they finally end up exploding that cell. And as that cell ruptures, it releases all those viruses again, and then each of those viruses go and find a new cell. So you can see how this can be very, very nasty. You go from one cell, takes over control, builds thousands and thousands of more viruses, and then those get all released when that particular cell explodes. This is why viruses can act so quickly and are so very dangerous. Bacteria, you get one cell, it divides, you get 2, then they divide, you get 4, then 8, then 16, then 32, and you can reproduce fairly quickly with bacteria. Viruses, takes one cell, infects one cell, that cell goes through this process, and then explodes, releasing 10,000 or more viruses. Each of those viruses goes and finds another cell, and so you go from 1 to 10,000 to 1 million to 1 billion very, very quickly. And not only does that happen, but as they reproduce, they cause those cells to explode into rupture, literally liquefying the tissues that they're attacking. This is why viruses like Ebola can act and kill within just a couple of days, and literally liquefy the entire internal organs of an organism, because they can go through this process very, very fast and spread throughout the entire body before the body can ever react. The other side of that is the lysogenic pathway. It's the hiding pathway. It's a very slow, methodical pathway that a lot of viruses take. And they inject their DNA or RNA, and then that material is actually put inside of whatever cell's DNA that's in there. In this case, it's a bacteria. In your case, it would be inside of your cells, like with HIV, inside of your cells that are with your immune system. And so that bacteria hangs out and reproduces. You produce more bacteria. Your cells divide and replicate, and you produce more cells inside your body. And every time it does that, it makes another copy of that DNA that's inside of that virus. And it keeps on doing that over and over and over again until it gets to a point where it starts to actually take over control of the cell, and then it reverts back to the lytic cycle. So it can hang out in this lysogenic cycle, going over and hanging out, waiting for more and more cell divisions until finally something triggers it, and they all come out all at once, killing off all the cells that they're infected in, and then going and attacking the entire body all at once. So rather than having one virus have to reproduce and go through and work through, you all of a sudden have thousands of them pop out all at once and attack the body and take it by surprise. So it's a pretty nasty little pathway. The other part to this is where they put their DNA inside of your DNA. They don't care. They just simply insert it into a specific location, and they don't care if they break or damage the DNA of that cell. So therefore, they can actually, a lot of times they implant it in areas that can lead to cancer. This is how you can get things like HPV leading to cervical cancer, is because it has this lysogenic cycle where it hides out in the DNA, and it literally ruptures and fractures the DNA in the locations that help to that normally help prevent cancer, and so therefore actually start leading to the mutations that cause cancer. So this is how viruses can actually end up leading to certain types of cancer. And we have some variations. We have something called reverse transcriptase, which is how the RNA functions to actually make DNA. So it's a specific type of protein inside of these guys that the RNA that allows the RNA to go back and actually create DNA, which can be put inside of the DNA of certain of the cells that they're attacking for the lysogenic cycle, and to take over control of that cell. You can also trick, like I said, they trick the cells to incorporate and to envelop those viruses and pull them in all by themselves without having to inject themselves. All kinds of different things. And so here's a nice little picture of how that happens. You can look at it. You don't need to know the specifics. And as far as infection versus disease, just make sure you know the difference between these two. Infection is when you get sick. Disease is when you're going to die. All right? Infection is when you've gotten something that's coming in, parasitizing you, making you sick, but you're able to fight it off. Disease is when that particular infection ends up overwhelming your body, and you cannot fight it off by yourself. You have to have help, and that's when it becomes a disease. So infection is you get sick, but you could fight it off eventually. Disease is you have no way of defeating it by yourself, and so therefore it turns into a function where it very well may kill you. Right? And hosts and pathogens are co-evolving all the time. Most pathogens do not want to kill their hosts, especially not quickly. Right? If they take their time, that allows them to be able to spread, to reproduce, and therefore the disease is able to procreate and continue on. And the flu is a great example. It's been around forever. It will always be around forever because you got an incubation period, and then you got a long period of time where you get people sick, and you very rarely kill people except people that are weak. Right? So therefore it's constantly spread between people because people keep on going and doing the things that they do, and it'll be around forever. You got things like Ebola. Ebola kills very, very quickly, causes very, very nasty symptoms very fast. Therefore, it's easy to stop as long as people are put in certain locations where they're not able to spread that disease, and you can identify them very fast because of how nasty it happens. Right? So you can quarantine people, keep them from spreading the disease, and it's much easier to shut down. This is why they've actually been able to overcome it in Africa, even though after it killed tens of thousands of people. It's because they finally got people to understand that if you leave the sick people alone, you won't get sick and this disease will go away. All right? But bacteria and viruses and infections in general don't want to do that because it tends to wipe out their disease, and you won't have it in the environment. The problem is that when it's infecting a novel host, so something for the first time. Ebola in us, this is the first time that Ebola has ever started attacking a human host. And so therefore, as it mutated and attacked us, it's very, very strong. We don't have any natural defenses against it. Same thing with HIV and AIDS. HIV transitioned over to humans from monkeys, and we have no natural defenses against an HIV virus. Several different species across the world have HIV within them all the time. They get it, and it's basically like a cold for them because they have a natural built-up immunity to that HIV after millions of years of them both co-evolving together. So that way the HIV still gets passed along, and it doesn't kill its host, and the host, it only basically makes it mildly sick. But since it's a new sickness to us, we had nothing to stop it, and it basically runs rampant throughout our body and kills us. So that's why we have diseases that are super dangerous, is because of this idea of a novel host, mutant strains, and these changes that prevent us from being able to prevent the virus from killing ourselves or the bacteria. Here's some main examples of ones that kill. So you got acute respiratory infections like the flu, kill over 1 billion people each year. Again, or have killed over 1 billion people per year, infect 1 billion people per year, kill over almost 5 million people per year. So you get a lot of people infected, not a lot of people dying, because most of those are going to be sick or very young or very old, something along those lines. You got the diarrheas, some really nasty like amoebic dysentery and things along those lines. Almost 2 billion people infected a year, 3 million die. Tuberculosis, 9 million infected, 3 million died. That's getting pretty strong. Malaria, 110 million, 2.5 million die. AIDS, 5.6, 2.6 million die. That's a lot. Half of the people infected die within a year. All right, measles, or die every year. Measles causes 1 million deaths a year. Unbelievable, since we actually have vaccines for measles, tuberculosis. And we still have this many people dying. Hepatitis B, tetanus, same thing. 500,000 people a year die from being infected, and only 1 million actually get the cases. And we still have a very, very cheap vaccine for this. It's amazing that this many people still die. And then we have antibiotic resistance now. So an upsurge in all these infectious diseases, and now these infectious diseases are now basically immune to the antibiotics we're able to use. We have MRSA, which is immune to a lot of the most common bacteria that we use. We actually have a antibiotic-resistant type of bacteria called VRSA that's resistant to vancomycin. Vancomycin is pretty much the strongest antibiotic that we have. You have to get it through an IV, and it is pretty nasty to your body as well as it is to the bacteria. But there are a type of bacteria now that are immune to that vancomycin. So there is no cure for certain types of diseases. All right? There are bacteria out there that we have no way of stopping because of this antibiotic resistance, because of people not taking the entire dose of their antibiotics, because of the antibiotics being spread throughout all of our food sources, because antibiotics are put inside of lotions and all kinds of stupid stuff like that. The antibiotics end up being distributed all over the place. You get bacteria developing resistances to them. You get those bacteria able to pass those resistances on to other bacteria, including infectious type of bacteria, and therefore we start having some serious problems. Now we're having a real issue with finding new antibiotics that are able to work and able to help us fight off disease. Now there are a couple of other infectious agents. These are kind of cool because they're very weird and kind of unusual. Right? The first one are viroids. Viroids are smaller than viruses because there is no protein coat. All they are naked strands of RNA. That's all there is, just the RNA. No protein shell, no tricking of getting inside the cell. They are just simply RNA. And they hide out and they go inside the cell's nucleus, interfere with gene regulation and making certain types of genes and making certain types of proteins. And they're a very common plant disease that can kill plants very quickly. But very interesting on how there's no protein shell, no injection, no tricking, just simply RNA little fragments that make their way inside the cell, inside the nucleus, and interfere with their gene regulation. And the other one we're going to talk about are prions. Prions are made from harmless proteins. All they are is the same proteins that are inside your body normally, but they have a very characteristic configuration, a very distinctive misfolding of these proteins. So these proteins take the normal proteins of your body and they cause them to fold in a very weird way. And this folding causes them to build up and causes them to interfere with certain processes in your body. And not only that, but they also go and they cause other proteins around them to misfold too. So they are infectious in that way, in that they are able to coerce other proteins to fold in the same way and cause problems again. So these misfolded proteins accumulate in the brain and in the nervous tissue and cause serious deterioration within those tissues. Now these things accumulate over time. As you get older, you get prions building up within your brain tissue, part of the degeneration of the brain. right, over time. Right? This happens. The problem is that we have certain things that have occurred that cause these prions to be able to be passed from an older individual to a younger individual. And when these make it inside of the high metabolism and high brain function of a normal individual, of a young normal individual, they go crazy. And they take over control, accumulate much faster, and deteriorate the nervous system of that individual very quickly, well, relatively quickly, and cause some really horrible side effects. And so the examples of these are Kuru and Creutzfeldt-Jakob's disease, which are two human diseases that were seen in groups that were cannibalistic. What some of these groups were doing is that when an elder in their tribe died, they would remove the brain from that elder. They believed that their power and their knowledge and their wisdom was kept within that, and they would cut it up into pieces, and everyone in the village would take a piece and eat a piece of that individual's brain, hoping to take in their power and their soul and their knowledge. Unfortunately, they actually ended up taking in some of those prions, and those prions would run amok throughout those younger individuals that ate the brain and cause this horrible disease. They cause a degeneration, basically brain damage and uncontrolled shaking and all kinds of horrible symptoms. We also saw this in animals. Back in the 90s, we had mad cow disease. In the 90s and early 2000s, we had mad cow disease. And so what happened, and we have scapies and sheep, is basically they took these animals, they slaughtered them, they took the remnants that we didn't eat, they ground it up, and they fed it back to those same animals again to allow them to get more protein. The problem is again, those prions are built up inside of all that nervous tissue that we don't generally consume, and that gets fed back to those organisms. So therefore, you get all these prions being built up inside of these different animals, and led to mad cow disease, led to this basically the same degeneration of their nervous system. Unfortunately, these prions from these animals could also get passed on to us because it built up in such large levels within those animals, and they were allowed to continue to survive and were actually slaughtered even though they were showing horrible signs of the disease. And then within that meat and within that tissue, there are some prions, and people actually would take those prions, would eat them, and then they would get sick. And so we had mad cow disease becoming a real problem in the 90s and 2000s. Luckily now they've learned their mistake, and we no longer do those practices, at least within most of the most of the first world countries. All right, so there's your prokaryotes. Make sure you take a look, try to narrow down a little bit of what exactly you need to know, and make sure that you're looking over this stuff so you're ready for your quiz. Have a good day. Bye.